Hi everybody. Let us discuss some of the questions from the second part of principles of inheritance and variations. So straight away enter into the question. First question. In which of the following derived crosses between true breeding lines the offspring neither genotypically nor phenotypically resembles either of the parents? Round green seeded, wrinkled yellow seeded. Round is dominant, green is recessive. Wrinkled, recessive, yellow is dominant. So naturally, in the F1 generation, the offspring will be round and yellow seeded. Means round and yellow seeded neither resembles phenotypically the parents nor genotypically. When phenotypically it is different, automatically it is genotypically too different. So first option must be the correct one. Let us see the answers. Let us examine the answers. Answer 1. So answer 1 must be the correct one. Let us see explanation for all this. In brown green seeded plants crossed with wrinkled yellow seeded plants, the offspring is phenotypically round and yellow, as I said earlier, and genotypically capital R small r, capital A small y. So genotypically it is different, phenotypically also it is different. In round yellow seeded cross with wrinkled green seeded plants, the offspring is round yellow seeded with the genotype capital R small r, capital A small y, double heterozygous here also. But here it resembles phenotypically the one of the parent, that is round yellow seeded. The genotype is different, but phenotype is same. Coming to the next one. Tall round seeded, dwarf wrinkled seeded are found naturally in a cross between tall round seeded plants and dwarf green seeded plants. The offspring has the tall round seeded plants. So it resembles genotypically, phenotypically neither of the parents. Green parts with round seeded, yellow parts with wrinkled seeded. The green parts with round seeded, yellow parts with wrinkled seeded. Automatically it will be green and round seeded it is. Green round. So green and round. Green round resembles the parent. One of the parents it resembles. Right? So genotypically it is different, but phenotypically resembles the green round seeded one. Coming to the next question. What is the phenotypic ratio of F2 generation of Mendelian derived cross? It's straight question. Phenotypic ratio of Mendelian derived cross 9:3:3:1. The straight one. The other ratios are entirely different. The fourth option must be the correct one. Fourth one is given. The phenotypic ratio of F2 generation of Mendelian derived cross is 9:3:3:1. Coming to the next question. In a Mendelian derived cross, the probability of formation of double heterozygous individuals in F2 generation is 1/4. Absolutely true. 4/16 means 1/4. Absolutely true. In the Mendelian derived cross, the probability of formation of double dominant phenotypes in F2 generation is 9/16. Yes, absolutely true. Because out of 16 that are formed, 9 will be double dominant phenotypes with 4 different types of genotypes. Right? So this is also correct. Both statements 1 and 2 are correct. The fourth option must be the correct one. Let us see the answer. Fourth one. In a Mendelian derived cross, the probability of formation of double heterozygous individuals, capital R small r, capital A small a, with respect to shape and color of the seed, and double dominant phenotypes round and yellow seeds, F2 generation is respectively 1/4 and 9/16. Coming to the next question. The best cytological evidence for Mendel's law of independent assortment is N of is 1. During N of is 1 at metaphasic plate, the alignment of maternal and paternal chromosomes are one bivalent, different from that of another bivalent. Right? Or other bivalents. It should be N of is 1. Let us see the answer. Second one. The best cytological evidence for Mendel's law of independent assortment is N of is 1. Separation of a pair of contrasting traits present on a homologous pair of chromosomes is independent of segregation of another pair of contrasting traits present on another pair of homologous chromosomes. Coming to the next question. Assertion reason. In F2 generation of hybrid cross, 2 types of phenotypes and 6 types of genotypes of formed organisms are seen neither in FP nor in FN generation. So, a little bit carefully how to read and understand it. What he wants to say is 6 types of genotypes are not found in parental generation, FN generation. Right? And 2 types of phenotypes are not found in FN generation, parental generation. Right? The Mendelian recombinant, 2 types of phenotypes. So 6 types of genotypes. Out of 6 types of genotypes, 4 types of genotypes belong to Mendelian recombinants, and the remaining 2 types of genotypes belong to the double dominant. So 4 types of genotypes are there for double dominant in F2 generation. So whose genotypes neither seen in the P generation nor in the FN generation? Assertion is correct. Inheritance of a pair of contrasting traits of a character is independent to the inheritance of another pair of contrasting traits of another character. Absolutely true and clearly explains. It is. So A and R are true and R clearly explains. So what option must be the correct one? Let us see the answer. In F2 generation of hybrid cross, there is a formation of 2 different types of phenotypes with respect to seed color and shape. So I have taken an example here, shape and color of the seed. Round green, wrinkled yellow. Right? So round green and wrinkled yellow entirely different from parental and FN generation. 6 different types of genotypes. Capital small r, capital Y, that is for round and yellow. Capital Y, small y, that is also round and yellow. Small r, capital Y, wrinkled and yellow. Small r, capital Y, wrinkled and yellow. Capital R, small y, round and green. And capital R, small y, round and green. So 2 genotypes are round and green, 2 genotypes are wrinkled and yellow, and 2 genotypes which are neither seen in the parental generation nor in FN generation are capital R, capital Y, capital Y, small y, that is for round and yellow seeded. So the 2 other genotypes for this round and yellow, one is the seen in the parental one, that is capital Y, round and yellow, and FN generation double heterozygous, capital R, capital Y, small y. So that is explanation for it. Coming to the next question. So this is here also it is given why it is so. The reason is given. It is because the transmission of round and wrinkled traits for the seed shape is independent of yellow and green color of seeds. Similarly, the transfer of R and R alleles for shape of seed is independent to transfer of Y and Y alleles to F2 generation of hybrid cross. Coming to the next question. Who among the following is not involved in rediscovery of Mendel's results? Correns is involved. Sharma involved. Davis involved. Sutton is not involved. Sutton and Boveri proposed chromosomal theory of inheritance. Both one is the correct one. So Sutton was not involved in discovery of Mendel's results. He proposed chromosomal theory of inheritance along with Boveri. Correns, von Sharma and Davis rediscovered Mendel's results. Coming to the next question is the matching. Carefully you have to see the phenotype is given in the list 1 and genotypes are given in the list 2. You have to match it. Round and yellow seeded, capital Y. Round and green seeds. Capital R, small y. So A3, B4. So wrinkled and yellow seeded. Small r, capital Y. That is C. That is 2, C2. Wrinkled and green seeded. That is small r, small y. That is 1, D1. So A3, B4, C2, D1. First option must be the correct one. Let us see the answer. Here it is explanation. Small r, small y, wrinkled and green. Capital Y, round yellow. small, capital, wrinkled and yellow, capital, small y, round green. Coming to the next question. Choose the incorrect statement. You have to choose the incorrect statement. Chromosomes occur in pairs. It is correct according to chromosomal theory of inheritance. Genes occur in pairs. This is also correct as given in chromosomal theory of inheritance. Behavior of chromosomes is parallel to the behavior of genes. It is also given in chromosomal theory of inheritance. So 1, 2, 3 are correct statements. Naturally, the fourth one must be the incorrect one. Alignment of a pair of chromosomes at metaphase plate is dependent on alignment of other chromosomal pairs of bivalence in anaphase. No, it is independent, not dependent on alignment of other chromosomal pairs of bivalence in anaphase. Right? That is anaphase 1. Right? So fourth option must be the incorrect one. Let us see the answer. Fourth one. The alignment of a pair of chromosomes at metaphase plate of a bivalent at anaphase is independent of another pair of chromosomes of other bivalents. It is anaphase 1. Anaphase means each group of haploid set of chromosomes moving towards a pole in anaphase 1 has various combinations of maternal and paternal chromosomes. That is, usually not all are maternal or paternal in a group. Coming to the next question. Basis for variations produced. What you identify, correct statements you identify. Basis for variations produced through sexual reproduction was discovered by T.H. Morgan while verifying chromosomal theory of inheritance. The statement is given even in the book. That's right statement it is. Correct, A is correct. Morgan worked with fruit flies to experimentally verifying chromosomal theory of inheritance. This is also correct. Morgan carried out the hybrid crosses in Drosophila melanogaster. This is also correct. A, B, C are correct. Morgan observed the independent segregation of alleles or genes for eye color and body color in Drosophila. Wrong. Not independent segregation. Together, they are inherited. That's why several parental combinations are obtained in F2 generation. In the hybrid crosses done in Drosophila with respect to body and eye color, 9:3:3:1 ratio of progeny was obtained in F2 generation. Absolutely wrong. That ratio was not obtained, deviated from this ratio. Whatever he obtained in F2 generation. D and E are incorrect. A, B, C are correct. So third option must be the correct one. Let us see the answer. Third one is given. While verifying chromosomal theory of inheritance by carrying out hybrid crosses, Morgan discovered the basis for variations formed thus due to sexual reproduction in Drosophila melanogaster or fruit fly. Morgan observed non-independent aggregation of genes for eye and body color in Drosophila, and F2 ratio deviated from Mendelian 9:3:1 ratio. Coming to the next question. Morgan coined the term. Morgan coined the terms linkage. Morgan coined the term recombination. Morgan also coined the term crossing over. So, he has not coined the term test cross. That was coined by Mendel. Not coined reciprocal cross. So, not coined hybrid cross. Only linkage. Second one is the answer. Straight question this is. Morgan coined the term linkage for physical association of two or more genes on the same chromosome. Eye color in case of fruit fly. That is linked genes. Genes for body color and eye color, linked genes. The linked genes on X chromosome. Coming to the next question. When yellow-bodied and white-eyed Drosophila was crossed with brown-bodied and red-eyed fruit fly, whose genes are X-linked, the result in F1 generation was. So in F1 generation, one obtained yellow-bodied and white-eyed males were obtained, brown-bodied and red-eyed females were obtained. Females were heterozygous in F1 generation. Because they are X-linked, they are. So on Y chromosome, these genes are absent. Right? So all males were red-eyed and brown-bodied, and all females were yellow-bodied and white-eyed. No, this was not the correct one. All males were yellow-bodied and white-eyed. This is correct. And all female were brown bodied and red eyed. Third option must be the correct one. Let us see the answer. Third one. Since the genes were body were brown dominant over yellow and eye red dominant over red color or X-linked in Drosophila, and it shows XY pattern of X-linked inheritance. In FM variation, all females that are heterozygous are brown bodied and red eyed, and males yellow bodied and white eyed. Coming to the next question. Setting organ is a unit used for chromosomal mapping. Third question. First discovered by Stuart Went. First he discovered how he established X chromosome gene mapping. Mapping of X chromosome done by Stuart Went. So straight question this is. Septum or scenting organ is a unit used for mapping the chromosome. It is nothing but percentage of recombination frequency. Third option must be the correct one. Third one is given. Scenting organ is a unit for recombination frequency used for mapping the chromosomes to understand the distance between genes and their sequential arrangement of the chromosome. This is the formula for that. One scenting organ is 1% recombination frequency. Recombination frequency is measured with the help of the formula number of recombinants formed divided by total number of individuals formed in a test cross into 100. Coming to the next one. Alfred Stuart Went was the first to do chromosomal mapping. Absolutely true. Chromosomal mapping is used as starting point to sequence the genome of an organism. Absolutely true. Both statements are straight statements. Linkage in the last paragraph they are given. Statement 1 and statement 2 are correct. The fourth option must be the correct one. Let us see the answer. Fourth one. Chromosomal mapping was first done for X chromosome of Drosophila by Stuart Went. It is used as starting point for sequencing the genome of any organism. Next one. This is explanation for the sequence of the linked genes. The recombination frequency with respect to genes W and Y is 1.3% by W and M. So here W for color of the eye, Y for color of the body, M for length of wings. So W at 1.3%, WM 37.2%, and Y and M 38.5%. So more is the recombination frequency, farther is the distance between the two genes. So here highest is Y and M. So Y should be kept one end of the chromosome, M should be kept other end of the chromosome. So in between Y and M there should be W. Moreover, the recombination frequency with respect to Y and W is very less. So W must be very near to the Y. So the sequence should be Y, W and M. So even the diagram will be clearly shown. So between Y and W should be 1.3%. Unfortunately 1.5 is written. 1.3% that is. W and M 37.2% and Y and M 38.5%. So the option sequence must be Y, W, M, or you can also M, W, Y, but M, W, Y is not there here. Y, W, M is there. So third option must be the correct one. Let us see the answer. Third one is correct. Here is the solution. Genes Y and M are the farthest as the recombination frequency 38.5% or 38.5 centimorgans. Gene Y and W are very close, and W is present just after Y as the recombination frequency 1.3% or centimorgans. Gene W is farther to M than gene Y, but present between Y and M genes as the recombination frequency with respect to W and M genes is 37.2%. Therefore, the sequence of these genes as chromosome Drosophila is Y, W, M. That is the explanation. Coming to the next question. Assertion reason. Male Drosophila shows crossing over with respect to genes for body color and eye color. Absolutely true. No, this is wrong. Male Drosophila cannot show crossing over with respect to genes for body color and eye color because it has got only one X chromosome. right? So Y chromosome is inherited the genes. When only one chromosome is there, there is no crossing over. The male Drosophila shows crossing over with respect to genes for body color and eye color. This is wrong. The assertion is wrong. Genes for body color and eye color are present on X chromosome. Yes, absolutely true. That is why male cannot show, whereas females can show crossing over during meiosis. So A is incorrect, R is correct. First option must be the correct one. Let us see the answer. First one here. In Drosophila, genes for body color and eye color and length of wings are present only on X chromosome, and Y chromosome lacks it. Since males have only one X chromosome, there is no chance for crossing over between X and Y chromosomes. In females of Drosophila, crossovers with respect to Y, W, and M genes take place because they have homologous chromosomes for these genes. Coming to the next question, again matching. Polygenic inheritance. Where polygenic inheritance is skin color in human beings, even height of the human beings. Also polygenic inheritance. So polygenic inheritance is also influenced by environment that you know. Pleiotropy. Here, mutation in gene that codes for phenylalanine hydroxylase. Best example of pleiotropy is phenylketonuria. So there is deficiency of malformation of phenylalanine hydroxylase. So B1, A2, B1. Linked genes. Linked genes for body color and eye color in Drosophila are linked genes. Genes that are not linked means that are present on different chromosomes. Genes for shape of pond and shape of seeds in P. So A2, B1, C4 and D3. First option must be the correct one. Let us see the answer. First one. So polygenic inheritance, skin color in humans influenced by three types of genes A, B and C. Pleiotropy, gene mutation in the gene for physical phenylalanine hydroxylase causes mental retardation, reduction in hair and skin pigmentation. Body color and eye color genes of Drosophila are X-linked. Pod shape and seed shape of genes of P are not linked genes. Which among the following genotypes show similar phenotypes in P with respect to seed shape and color? So, among the four genotypes, which one show the similar phenotypes among the genotypes? Right? Catlar, catlar, catloi, catloi. Right? Catlar, catlar, catloi, catloi. Round and yellow. Catlar smaller, catloi small y. Round and yellow. It is also round and yellow. Catlar, catlar, catloi small y. It is also round and yellow. Capital smaller, capital small, round and yellow, like B, same, B repeated here. So, all the four are same, right? So, A, B, C and D. Fourth option must be the correct one. Let us see the answer. The phenotype round and yellow seeds has four types of genotypes. They are Caplar, Caplar, Caploi, Caploi, Caplar smaller, Caploi, Caploi, Caplar, Caplar, Caploi small y, and not be both are one the same, that is capital R, small r, capital small y. So these are four types of genotypes, but there which is not mentioned, capital R, small r, capital Y is not mentioned there, but here in the explanation I have given, there B and D are same. Anyhow, it is A, B, C, D. Right. Coming to the next one. Here again, linkage. Choose the correct statement with respect to genes W, Y and M linked on X chromosome Drosophila. So from the diagram very clear that Y and M are weakly linked, means distance is more. Y and W are strongly linked. W and M are relatively weakly linked compared to Y and W, but strongly linked compared to Y and M. That Y, W are strongly linked compared to Y, M and WM. right? And WM are strongly linked compared to YM, but weakly linked compared to Y and W. You know the statement that more is the distance between the genes, loosely they are linked. Weak is the linkage. Very nearer the genes, strongly they are linked. Higher the recombination frequency, weak linkage. Lesser the recombination frequency, strong linkage or strongly linked. Based on that, it is, let us see the statements. Genes M and Y are tightly linked than W and M. Y and M not tightly, this is wrong. Incorrect statement. Genes M and Y are tightly linked than Y and W. Now this is also wrong. A, B are wrong. Naturally C and D must be the correct ones. More crossovers are same with respect to Y and M. Yes, absolutely true. Farther is the distance, more are the crossovers than W and M. Yes, absolutely true. More crossovers are same with respect to W and M. Absolutely. Than Y and W. Absolutely true. C and D are correct. So second option must be the correct one. Let us see the answer. Second one. Genes Y and M are loosely linked than W and Y and W and M. Genes Y and W are tightly linked than W and M and Y and M. Very from the figure also we can clearly understand. Therefore, chances for crossing over is more between Y and M than Y and W and M. It is very less with respect to Y and W compared to Y and M and W and M genes. Coming to the next question. Which among the following are recombinants with respect to eye color and body color in Drosophila? Which are recombinants? That are formed in F2 generation? White-eyed and yellow-bodied flies, they are not recombinants. They are true breeding. Red-eyed and brown-bodied flies, these are also true breeding, they are not recombinants. Red-eyed, yellow-bodied flies, yes. Red is dominant, yellow is recessive. Brown is dominant, brown body is dominant, but yellow is recessive. So one dominant, one recessive, it is recombinant. White-eyed and brown body is also recombinant. C and D are correct. Second option must be the correct one. Second one. White eyes and yellow body are the recessive features of Drosophila, whose genes are present on X chromosome. Red eyes and brown bodies are dominant traits of Drosophila. These individuals may be either homozygous or heterozygous in females. The recombinants for these characters would be yellow body, red eyes, and brown body and white eyes. Next question. In the genotypic ratio of F2 in a diploid cross of P with respect to seed color and shape, which phenotypes have only two types of genotypes? So, double dominant phenotype, four types of genotypes. Double recessive, only one genotype. Whereas the recombinant, they are round and green, wrinkled and yellow. They have got two types of genotypes. So B and C have got two types of genotypes. Both option must be the correct one. See here. Genotypes are round and green. Capital R, capital Y, small y, capital R, small y, round and green. Genotypes for wrinkled and yellow, small r, capital Y, small r, capital Y, small y. Genotypes are round and yellow. Four types of genotypes are there. Not two types, four types of genotypes are there. So, the capital R, capital Y, capital Y, capital Y, small y, capital R, capital Y, and capital R, small r, and capital Y, small y. Four types of genotypes for double dominant phenotypes. So, this is double recessive. Only one genotype. Phenotype 1, genotype 1. Genotype wrinkled and green. Small r, small y. So, double recessive, only one genotype for the phenotype. Double dominant, four genotypes. And recombinant, two types of genotypes for each. So, thank you very much. That is all about the questions in the remaining portion of principles of inheritance and variations. Thank you.